Hello, everyone, and welcome to the eOrganic webinar on raw milk parameters that influence pasteurized dairy product quality. So today I'm very pleased to welcome Dr. Nicole Martin, who is the Associate Director of the Milk Quality Improvement Program, or MQIP, in the Department of Food Science at Cornell University. She studies the transmission of spoilage organisms into raw and processed dairy products, and she develops strategies to reduce or eliminate this transmission. Funding for this webinar is provided by a NIFA OREI grant that is looking at ways to improve the quality and shelf life of organic dairy products. So with that, I'm going to hand over the screen control to Nicole Martin.
Okay, I think I have control. Wonderful. Thanks so much, Alice. I appreciate it. And thank you all for joining us today. It's afternoon here in New York State, but morning for those of you on the West Coast. As Alice said, my name is Nicole Martin. I'm the Associate Director of the Milk Quality Improvement Program in the Department of Food Science at Cornell. We currently have a USDA NIFA OREI grant, and we are studying the quality of organic raw milk and organic finished dairy products. And today I'm going to spend our time talking about how raw milk parameters impact pasteurized dairy product quality. Before I get started, I do want to acknowledge that Cornell University is located on the traditional homelands of the Guayacona, the Cayuga Nation. The Guayacona are members of the Haudenosaunee Confederacy, an alliance of 6 sovereign nations with a historic and contemporary presence on this land. The Confederacy precedes the establishment of Cornell University, New York State, and the United States of America. We acknowledge the painful history of Guayacona dispossession and honor the ongoing connection of Guayacona people, past and present, to these lands and waters. So first of all, I think big picture, what I can say, if I had to distill today's entire talk down to one sentence, what I would say is that what happens on the farm does not stay on the farm. So pasteurization is not a magic bullet. And I run into a lot of folks in my work who have sort of this conception that why does it matter what happens on the farm or what the quality of raw milk is? We're going to pasteurize that product and it's going to be okay in the end. And really, that's truly not how it works. And so my goal today is to introduce you to these parameters that really have an impact on finished product quality, and convince you that high quality raw milk really is essential to not only manufacturing high quality processed dairy products, but ultimately affecting how consumers perceive the quality of organic dairy products. So we'll cover the sort of major factors, the major parameters. Not all factors, not all raw milk quality tests have an impact on finished product quality. And we'll talk about some of the key testing that should be done to determine product quality. So just quickly, there are 4 big parameters that we'll talk about today. The first being somatic cell count, which many of you are probably very familiar with. Total bacteria count, spore count, which might be a new subject for some of you, but this is really the focus of our OREI grant, our spore forming bacteria, and I'll introduce you to those. And then finally, raw milk odor and flavor profiles and how those can impact finished product. So, and then when we're thinking about those 4 parameters, then there are multiple factors at the farm that influence those parameters. So there might be cow level factors or sort of environmental factors, the milking time hygiene or milking time factors. And then finally, there's some factors that happen actually at the processing facility, but influence those raw milk parameters. So we'll talk a little bit about those as well. This is our dairy plant at Cornell University, by the way. It's got lovely cow print pattern on the wall, so it's fun to visit. So let's start out with somatic cells, probably one of the most well-recognized raw milk parameters. So somatic cells have a well-established known impact on finished product quality. These are white blood cells. They're produced as an inflammatory reaction. This is commonly called mastitis, and often that's in response to a bacterial infection. Variation in levels of somatic cell can be associated with non-infection factors like stage of lactation, parity, or how many calves the cow has had, and other intrinsic cow factors. The U.S. Pasteurized Milk Ordinance limit for somatic cell count is, of course, 750,000 cells per mil, which is way, way above what we would expect for the sort of typical herd or even typical cow. So an average healthy non-infected quarter somatic cell count is around 70,000 cells per mil. And then typically the benchmark of 200 to 250,000 cells per mil is used for an indicator of mastitis. And here I just have a plot of the milk weighted and producer bulk tank somatic cell over the last 20 years or so in the US. And you can see that there's just been a precipitous decline in the sort of average bulk tank somatic cell over time, from around 300,000 in the sort of 2000s, early 2000s to around 200,000 in 2018. So major improvements. Producers do an excellent job managing for somatic cell, and this is obvious in these trends. When we think about what happens with a finished dairy product in product that has elevated somatic cells, a lot of the data that we have, a lot of the evidence that we know of comes from cheese. So somatic cells can impact both yield as well as quality of cheese, even fermentation efficiency, so how well that milk turns into cheese. So we know when there's an increase in somatic cell, we get reduced yield, and that's really driven by an alteration of the composition of that raw milk. So we see decreases in casein, lactose, and calcium, and increases in albumin, sodium chloride, and various enzymes. And those enzymes are really important because they are released when the somatic cells break down in the raw milk. And the one that we are really interested in here, plasmin, is actually a heat stable protease that can survive pasteurization or survive different heat treatments during processing. This table I have here is just an example of the comparison of different raw milk components in normal milk to high somatic cell count milk. And you can see some of them are reduced, like lactose, and some of them are very high compared to the normal milk, like lactoferrin. So these all contribute to what we see in the finished product. So when we have those increases in somatic cell counts in the raw milk, we see this increased clotting time. I was talking about the fermentation efficiency, so it takes longer to make cheese. Increased moisture, reduced yield when we correct for that increased moisture, so we have less actual protein in that product. Reduced fat levels, and then we can see flavor and body defect development due to these heat stable enzymes that are left over in that milk. This plot that I have here on this slide shows, if you want to look just at the bottom one, which is a total sensory score when you compare cheese made out of low somatic cell count milk and medium and high somatic cell count milk, you can see that once you get to that high category, which in this case I think was somewhere around 500,000 cells per mil, so quite a bit above where we would see the average US bulk tank somatic cell count, you really see those significant reductions in the flavor and total sensory scores. So an inferior product altogether. When we think about what at the farm level influences somatic cell count, this is really focused, of course, at the cow and environment level as well as the milking level. I won't go into this in incredible detail, first of all, because I'm only here for 50 minutes and I could talk about things all day, but I'm not an expert in somatic cell count. Just as a little bit of a teaser, we're going to have a workshop in December where we'll dig into all of these factors in depth with experts in each of the areas that I'm talking about today. And we'll really get into how to manage somatic cells at the farm level. But just as sort of an overview, of course, we know that things like the type of mastitis pathogens that are causing this elevation in somatic cells, like Staph aureus, they can cause this sort of chronic increase in somatic cells. The season can impact somatic cell levels. Milking practices and hygiene, like wearing gloves or pre-dipping and post-dipping, and what you use for those materials. Milking equipment factors. Are there automatic takeoffs on the milking clause? Are you milking in low flow? Are there rubber issues or preventative maintenance issues? So those types of things. And then obviously when the cows are in a housing area, or what type of housing area there are, and the cleanliness of their environment have an impact on somatic cell. The next parameter that I'm going to touch on is total bacteria count. And we know this parameter by a couple of different names. So we might call this a total bacteria count. We might call it a standard plate count or a total viable count. The PMO limit, of course, is 100,000 CFU per mil for individual producers and 300,000 for commingled raw milk. Most farms can easily achieve 10,000 CFU per mil if everything is sort of going right. And that's a limit or a measurement of quality that can be used. When we think about a total bacteria count, we're actually using a few different subsets of bacterial populations that make up that entire population. And the four big ones here are psychrophilic bacteria like Pseudomonas. These are organisms that grow at low temperatures. Thermoduric bacteria like Micrococcus or certain staph and strep that can survive minimum pasteurization temperatures for vat pasteurization, or sometimes called a laboratory pasteurization count. Environmental contaminants like coliforms, and mastitis causing bacteria like Streptococcus. So when we think about all of those different populations, we need to really narrow our focus onto which of those populations can actually impact finished product. And when we do that, we're really focusing on the psychrophilic bacteria like Pseudomonas, because they can grow at low temperatures. And that's just demonstrated here in this little plot that I have, where we look at temperature here on the x-axis and growth rate of bacteria on the y-axis. It's just important to know that as the curve increases here, they grow faster. So for psychrophilic or psychrophiles, they prefer low temperatures, so refrigeration temperatures. Mesophiles prefer sort of ambient temperatures. And then we have the thermophiles and sort of the extremophiles, which grow at really, really high temperatures. And so when we have either a high initial contamination with these organisms like Pseudomonas, or we have improper cooling or very long hold times, even with proper cooling, we can get high levels of these psychrophilic bacteria. And they can even exceed the PMO limit by the time that milk gets to the processing facility and is actually processed. And this is important because a lot of these psychrophilic bacteria like Pseudomonas, once they reach those high concentrations, typically, greater than 1 million CFU per mil, they can produce heat-stable enzymes. These are different heat-stable enzymes than the somatic cells that are associated with the somatic cells, but they function similarly in that they can survive pasteurization and then go on to degrade the proteins and the fats in the product over the shelf life of the product. Most of them, like I said, don't do this until they reach about 1 million cells per mil, but some strains can do this at around 10,000 CFU per mil. So there's variation depending on which strain is in the product. These heat-stable proteases, so enzymes that can break down proteins or enzymes that can break down lipids or fats, which are lipases, they can lead to reduced yield, same type of issue that we see with the somatic cell enzymes. Some estimates of 3 to 4% reduction in yield. And then they can also influence flavor, odor, and even sort of the texture of different dairy products. So at the, we're talking about what happens at the farm that can affect the total bacteria count in raw milk. Certainly cow and environmental level factors, milking factors. And then this isn't a unique one, because what happens at the processing facility before pasteurization can actually influence this. And I'll touch on that just briefly. So first of all, what happens at the farm? So when we see SBC counts or total bacteria counts exceeding about 10,000 CFU per mil, remember I use that as sort of the achievable quality level for most producers. We can sort of break down what could be causing this increased somatic, or not somatic cell, total bacteria count above 10,000. So typically we would not expect the natural flora of the cow to be causing an increase above 10,000. It is possible that mastitis organisms could cause that. It is possible that dirty cows or dirty equipment or poor cooling could cause that. So there's a variety of things to look into when we see those total bacteria counts sort of increasing, both from a cow level perspective, an environment perspective, and then an equipment perspective. Now, if we see those counts exceeding 100,000, which again is the PMO limit, then what's most likely is either the equipment is dirty and the milk is getting inoculated essentially with the bacteria that's growing in the equipment, or there's poor cooling and the bacteria that are there are just growing quickly to very high levels. So there's some ways of sort of some diagnostic tools, I guess, to breaking down how these different levels are possible. And then I do really, again, just really quickly want to touch on the fact that the processing facility can influence this raw milk total bacteria count, because how that raw milk is handled can influence those counts. Bacteria are living, growing organisms. They're not like somatic cells, which sort of what's in there is what's in there. Sometimes you can see a decrease depending on how the milk is handled, but they're not going to increase. They're not multiplying, they're not growing, but bacteria do. And so even if the total bacteria count is, good at the production level, if that product is either held for a long time or not held at the correct temperatures before pasteurization, then those bacteria can grow to levels that can cause these issues in the finished product during that time. And so it's really important, I think, for producers to understand that can happen, but also just to understand sort of the how no individual step in the dairy processing continuum and dairy production continuum is a lone island. They all sort of roll into each other and impact each other. So as I was saying, what happens at the farm, even if everything is great at the farm level, if that product is not handled correctly, afterward can cause issues in the finished product. And so what I encourage processors to do if they're going to hold their raw milk before processing is to just double check those bacterial levels as sort of a control point for making sure that we don't have reduction in yield for cheese or quality issues in different products like cheese and fluid milk before processing. All right, so let's move on to spore-forming bacteria. I'm going to spend a little more time on spore-forming bacteria because most people at the production level that I work with don't have a lot of exposure to spore-forming bacteria. So I just want to make sure that everybody's on the same page with me. So spore-forming bacteria are endospores. Here I have a micrograph image of a bacterial cell. That's this sort of outside cell here, and then the spore or endospore in the center. These spores are formed when a bacteria is under a stressful condition. So maybe it's run out of nutrients, or it's in a scenario where it doesn't have enough moisture or something like that. this special group of organisms can create these spores, and that allows this bacteria to persist in these environments that would otherwise be lethal. So, for example, when we have spores in raw milk, they can actually survive pasteurization. HTST, high temperature, short time, low temperature, long time pasteurization, even some that can survive UHT pasteurization. So these cells are extraordinarily resistant to heat. They survive just fine in powders, under different sanitizers and radiation. So they're very, very resistant structures. And then once that spore finds itself in a favorable environment again, so maybe in some fluid milk that's being held at refrigeration temperature, those spores can then return to their metabolically active state and start growing again. So these are really tricky to get rid of and really tricky to control in processed dairy products, and they come from the dairy farm environment. I like this image because it really shows how this bacteria can do these sort of two different life cycles. The first being what we call a vegetative cycle. That's just when the bacteria is sort of growing, it's dividing, it's metabolizing nutrients, it might be creating some enzymes, it's doing its thing. And then it, at some point it enters the sporulation cycle when it realizes that it's in an environment that's not favorable to growth. So again, limited nutrients, limited moisture, something like that. It goes through this series of steps, and this leads us to this, what we call a free endospore or spore. It's important to make a distinction that these are not mold spores. These are bacteria. And so it's definitely, not the same thing, not the same issue. But you might have heard the term spore before. But this spore can then re-enter this vegetative cycle at some point when it's in a favorable environment. So this is just a little video of these spores re-entering the vegetative cycle. So you see these bright spots, those are the spores. It's a time lapse. This doesn't happen really quickly. It happens over, even under really good conditions, the optimal conditions, it still takes several hours for this to happen. And you can see that some of these spores are starting to grow, and you can actually start to see them dividing. And this is how bacteria grow, right? 1 becomes 2, 2 becomes 4, and that's how we can end up with a lot of bacteria in a very short period of time. We have certain types of spore-forming bacteria that cause fluid milk spoilage. We have certain types that cause cheese defects. This is one that's created by anaerobic spore formers. So that's just the type of bacteria that does not grow in the presence of oxygen. It has to have a no oxygen environment, and causes a defect in cheese called late blowing. We call it late blowing because it doesn't happen until typically around 60 days into aging the cheese. And then all of a sudden there's a lot of gas production, there's a lot of smelly compounds produced in it, and it's essentially causes the destruction of the cheese. So here's the control cheese at the bottom, and then the cheese that's got these anaerobic spores. Clostridium tyrobutyricum is the organism that most of the time causes this defect, and these are just additional pictures of that defect. Mesophilic and thermophilic spores are important in dairy powders. So dairy powders for export and domestic consumption. So there's oftentimes very stringent limits on spores in dairy powders, and typically we see those being here less than 500 to less than 1,000 per gram of powder. And remember, this is a concentrated product, so the the level that's in the raw milk, it gets concentrated into this powder. And we see that there are 3 primary organisms that are in powder in the US and actually all over the world. This is consistent: Bacillus licheniformis and Oxybacillus and Geobacillus. Bacillus licheniformis happens to be the most common spore former found in raw milk. So there's a strong connection between the level of these spores in the raw milk and the quality of the powder that can be used for export or used for other purposes. So when we think about what happens at the farm that can affect these spore forming bacteria or the level of spores in the raw milk, we're mostly concerned about cow and environment factors and milking time factors. And I'll walk through some of the key parameters here. So I broke this down into three bins: udder hygiene, milking practices, and silage spore levels. So first, talking about udder hygiene, there are a variety of factors at the farm that contribute to udder hygiene. We know that udder and hygiene have a huge impact on the level of spores that we find in the bulk tank milk. And we know that things like overstocking causes higher levels of dirty udders, loose consistency of manure, which is of course tied to the cow's diet, how frequently bedding and grooming of stalls when the cows are inside, if they're inside, cleanliness of the areas the cows are walking in, how frequently they have access to outdoor areas. And of course, we know in organic production, that's a key parameter. And then sort of just general stockmanship factors like rushing animals. When cows are rushed, they splash more manure and soil, whatever they're walking in, onto their udder, and that increases the dirtiness of those udders. When we work with producers, we generally recommend that they do some udder hygiene scoring. And this here is just a scorecard from the University of Wisconsin where the cow is scored as either a 1, 2, 3, or 4 for udder hygiene, a 1 being very clean and a 4 being very dirty, right? When those hygiene scores all together, you look at your entire herd, you should have less than 15% of the cows having udder scores of 3 or 4, so those two dirtiest. If it's more than that, then it's important to go back to some of these management factors that affect udder hygiene and see where those things can be affected in order to improve the udder hygiene. As far as milking practices, we know that milking practices really are the bulk of the sort of influence on how many spores can get from the environment into the raw milk. And forestripping is sort of at the top of the pack for things that are important. Forestripping prior to unit attachment is part of your milking procedures. And that's really for two reasons. Number one, because between milkings, some bacteria can accumulate in the teat canal. And that's normal for bacteria to be able to get into that teat canal. But when you forestrip, you're removing the first few streams of milk, and those streams of milk kind of flush out those accumulated spores that are sort of just sitting there between milking. And we've done this work, and others have done this work where we look at the spore levels in that forestripped milk, and then in the next few streams of milk, and we see that there's significantly higher levels of spores in the forestripped milk. So that's part of it, is we're just physically removing some of the spores. But really, what is most likely causing the biggest association with lower spores in the bulk tank is that when you get that good stimulation, you get a unimodal milk curve. So you get good milk flow, good milk letdown, and you're not milking in low flow. And that's what's demonstrated here in these two plots. So here on the right is an example of milk flow over time. And this is a unimodal curve. So this is, one giant curve. And what you see is that the milk is, coming out very, very quickly, sort of plateauing, and then coming down quickly. And so your milking time is all in, above this 2 pounds per minute cutoff. When you don't have that good stimulation before you put on the unit, you get this bimodal milk curve that I'm showing here, where you get an initial increase in the milk flow, and that's taking out what is sort of in that milk cistern where about 20% of the milk is sort of just sitting there ready to come out. But then you don't get the actual milk let down until later on, and you get these two different peaks in the curve. The time that the unit is on below this threshold, below this 2.2 pounds per minute flow rate, is actually causing a lot of issues, not just sort of with how much milk is being milked out, how long it takes to milk out, but this can actually cause damage to the teat ends, increase the risk for mastitis, and cause calluses and damage to the teat end, which I'll show you in the next couple of slides, is also related to spores in the bulk tank milk. So for shipping or any other form of good stimulation is probably the most important thing, is getting that good milk letdown so that you preserve teat ends. And so moving on to teat end condition, this is a sort of cross-sectional micrograph of two teat ends. The one on the left here is a healthy, smooth teat end. The one on the right here is a damaged teat end with a lot of calluses, a lot of damage. You can see that the teat canal here, and right here at the end is this little sphincter that closes 30 minutes after milking. That muscle closes in order to prevent contamination of the teat canal with bacteria that are in the environment. So when the cow's, laying down in her bedding or on the pasture for 12 hours or whatever she's getting, the bacteria are not able to enter into this teat canal. With a damaged teat end, you can see that, the ability of that teat end to adequately close is significantly reduced. And so we have sort of this open pathway to bacterial entry. Not only that, but trying to clean off a teat end that's all rough and calloused is extraordinarily difficult. It's hard to get all of the material off of a teat end that is in bad condition. And so those two factors lead to higher spore levels in bulk tank milk, or from individual cows with damaged teat ends. And then we know that reducing kickoffs helps. That can really be tied to things like teat end condition, right? Because we know that cows that have lesions or damaged teat ends, that can be really uncomfortable to have to be milked. And so they're more likely to kick off. And that really is just about what is that milking unit falling into when it's kicked off. And then lastly, training parlor employees to fully clean teat ends, because a lot of times, parlor employees might be in a rush, or they might not have good lighting, and so they can't see the teat ends really well. There might be understaffing issues, and so they're trying to cover more cows. And so the ability of that employee to fully clean the teat end, and the willingness of that employee to fully clean the teat end and do that very consistently, we also know influences spores in bulk tank milk. In fact, we did a study where we did just this. We trained parlor employees on sort of really focusing on teat end cleanliness, and from before to after, we saw a 40% reduction in spore levels in the bulk tank, not on the individual cow level, but actually in the bulk tank. So this is a really big factor. And then lastly, I'll just touch on the silage spore levels, because this one has a really strong historical association with those anaerobic spore formers like Clostridium tibutyricum that I was telling you about, that caused the late blowing defect in the cheese where it sort of blows up. There's this historical association with those Clostridium tibutyricum and high levels of those organisms in the silage. And in fact, in some areas of the the world where the cheeses that are susceptible to this spoilage are made, like in Switzerland and the Netherlands, the milk used for those cheeses can't come from cows that are fed silage. That's in Switzerland. And in the Netherlands, they actually penalize levels of these particular spores in finished product. And that is tied back to the silage quality. All right, I'm going to wrap up with flavor and odor profiles of raw milk. And this is one I think that is less commonly tested, less commonly looked at, but certainly can have a huge impact on finished product quality. In fact, I have been working with a cooperative that the milk goes into fluid milk, HTST fluid milk, and they were having some customer complaints on a fresh product, oxidized flavors in fresh product. And we were able to identify which producers that oxidized flavor was coming from and resolve that issue so that they were able to eliminate their consumer complaints and eliminate that defect. So I think it's a huge issue, could potentially be a huge issue depending on the practices. And then, it's just not one that's, I think, commonly looked at. So off odors and flavors, so smells and tastes, they can originate from the farm environment. They can be absorbed, so they can actually be, if the cow is breathing in some odors, like, strong barn smells, those smells can be transferred from the bloodstream or from the lungs into the milk that way. They can be from bacterial contamination, so certain bacteria produce these off odors and off flavors that can persist even after processing. So the processing or the pasteurization doesn't get rid of them. They just kind of keep going afterward. Or they could be chemical. So they could be something like oxidized flavors that come from over agitation or exposure to certain metals or those types of things. So we know that some of the common defects, they're common odor and flavor profiles that we see in raw milk can be barny, cowy, feed and weed, and those are to some degree expected in organic products and not considered a defect. But if they become very strong, then they can become objectionable. Malty, which is a bacterial due to a bacterial contaminant, or like I said, oxidized, and actually oxidized seems to be the most common attribute that's found in raw milk. And like I said, most of these defects or most of these attributes actually will carry over into the finished product. The pasteurization does not eliminate these at all. The table I'm showing here is just from a study where they looked at the transmission of feed flavors based on what type of feed was fed, and then the amount of feed that was fed and the interval before that the cow ate the feed before milking. And you can see just sort of with this first alfalfa hay example that if 2 to 6 pounds of feed were fed, the cow is milked after 2 hours, there was an objectionable feed attribute. But if it was 5 hours before milking, then there's no residual feed flavor in that milk. So there's a lot of factors that can go into this, but certainly, the time, the exposure, those type of things can all have an impact. So I just wanted to wrap up with, examples or recommendations for doing some raw milk screening, both at the production level as well as potentially at the processing level. This can be done periodically, so it can be done annually or quarterly or monthly or even more frequently. This is sort of an involved process and takes some training of people in order to identify these different flavors, flavor and odor attributes. We recommend doing some sort of a heat treatment, typically a 63 degrees Celsius for 30 minutes, which is vat pasteurization or low temperature long time pasteurization, and then quickly cooling and then screening the milk with, just tasting, smelling. and having, a few people do this. We would recommend 3 trained individuals, but if that's not possible, even one person doing it is, gives you some feedback on if these flavor and odor defects are showing up. And then you can go back to the farm level and figure out what's going on that could be causing this defect. Okay, so hopefully, I know I covered a lot, very, very minimally. But hopefully I'm leaving you with the thought that what happens on the farm really does influence the finished product quality. And ultimately, in the dairy industry, we want consumers to be happy with our products. We want them to find them, high quality and good tasting and enjoyable to consume. And so having control of these management and management factors and parameters at the farm level is really important to producing those high quality dairy products. So I did just want to bring your attention to, again, this raw milk quality workshop that we have coming up. So this is actually, it's a free workshop. It's two part and it's virtual. And we're focusing on these parameters, what drives them, and then what management practices or factors at the farm influence them. And we'll have an entire hour-long talk on each of these different things so we can really dig into mastitis management and teat and udder health and milking system management and the housing area and bedding factors. So this will be in 2 parts, like I said. It's a 2-hour self-paced sort of raw milk quality 101. We'll go over what bacteria are found in milk, what somatic cells are, what are, sort of some common flavor and odors in milk. And then that will be followed by a 5-hour live virtual workshop on December 16th. So the first one, the shorter one, is self-paced. It can happen anytime in leading up to the live virtual workshop. And then the live virtual will be in person, and we'll have our speakers doing that. So keep an eye out for registration details. They'll be coming through the eOrganic listserv soon, probably in the next few days. We're not limiting registration, but we do have some interactive components of the self-paced workshop, and those will only be available to the first 50 participants because, we only have enough kits to send out. We're actually going to be shipping out interactive kits to go to the first 50 participants. So that's just something to keep in mind if you're thinking about doing it. When those registration details come out, it's probably a good idea to go ahead and register so we can get those materials to you. Okay, and that's all I have, and I'm happy to answer questions for anybody who has any.
Okay, that's great. I'm going to turn my video back on here. Thank you, Nicole. I'm going to put the information while Nicole is answering a question of how you can sign up for the eOrganic newsletter to get the information about that. Basically, you go to eorganic.info and then you can sign up for our newsletter. So let me just pull up the Q&A box. I know we've had a few questions here. The first one was, so I was just curious how you count somatic cells in milk. Is that, do you have a particular device that does that? Some kind of automated cell counter?
Yeah, great question. So most somatic cell counting is done with a technique called flow cytometry. You might have heard of a Bacto scan before. And essentially what happens is the somatic cells are sort of fed through this very fine capillary tube. And as they are fed through that tube, they sort of line up individually in a row. And then as they move through that capillary tube, they move past a laser, and that counts the somatic cells. So very, very fast technique. It can be done in, a couple of minutes. So you get quick turnaround. There are some folks who are still doing a microscopic technique, so a direct microscopic somatic cell count. And that technique is also, very fast because it's microscopic, but it's not as automated, right? You have to have a human who is trained to do it. There are some particular, equipment that you have to use and some calculations, but, it's certainly still possible. And I know folks who still do it that way as well.
Okay, great. Okay, so this goes back to the earlier part of your presentation. Why the different SPC limits for single load 100K versus commingled loads at 300K?
Yeah, that's a really great question. So this really comes back to the fact that once the milk leaves the bulk tank and gets commingled into the truck, that that route might be different lengths. You expect some amount of bacterial growth once that process starts. So once it gets to the receiving point, and they check that level, it could be a little bit higher. So it's allowing for some bacterial growth after that point. When you have different milks coming in together, again, you might have this, one's a little bit higher than another. And so it allows for some flexibility with that.
Okay, great. Let's see here. Someone says, thank you for the great presentation. Have you studied the effect of microfiltration of raw milk to eliminate or reduce the amount of the bacterial spores and therefore extend the shelf life?
Yes, this is an ongoing area of research for us and for some of my colleagues here in the food science department. So microfiltration is quite effective at eliminating or removing spores. We see typically a 1 to 2 fold or 1 to 2 orders of magnitude reduction in the level of spores, which can extend the shelf life of products quite considerably. This technique can also be used for cheese makers who want to reduce the level of those anaerobic spores that come into the process to prevent that late blowing defect. So the tricky thing with spores is that as few as 1 spore, for example, a cyclotolerant spore or a spore that grows at low temperatures, 1 in a gallon of milk can cause shelf life issues over 20 days. Common shelf life for fluid milk is around 17 days. Sometimes it's as low as 14 days. So even very, very low levels can cause this spoilage in fluid milk. And then for cheese, as few as 1 per 100 mils can cause spoilage. So we're talking about extremely low levels. So microfiltration absolutely can help, and that's being used, in some areas to improve shelf life. But it's certainly not a magic bullet. Nothing is a magic bullet, unfortunately. So it can be a really useful tool, though.
Okay, great. What is your recommended method for screening SCC on the farm? Is it CMT or mass detect or something fancy with a cell phone? That one.
So that's a really great question. So a lot of the producers that I work with use CMT, and that is a really easy and fast tool for screening. Obviously just for stripping and looking at the milk is a good tool for screening for potential issues. I'm not as familiar with any new, potentially digital tools that are out there. I know that there's some systems that will measure and screen for SCC, but I don't have a lot of good details on those. That's something that we can dig into, though, at our workshop. So we'll have an entire hour with someone who's, just an expert in mastitis management. So please attend the workshop, and we can dig into that further there.
Okay, great. Let's see. Any suggestions for handling oxidizing flavors? We are seasonal 100% grass-fed, and when we start spring milking, there can be a strong flavor from sudden extra omega-3. Not sure if that is oxidizing. It clears up quickly, and we use DHIA for testing. And I think it might also be interesting to just hear what you mean by oxidizing or oxidized flavors.
So oxidized flavors can be a sort of dumping ground for a lot of different things, and there are a lot of different mechanisms for oxidized flavors. So certainly the types of fatty acids in the milk are going to cause differences in oxidized flavors. But if from the perspective of other issues, so there's oxidation that can happen because of exposure to different chemicals or metals. There's oxidation that can happen because of over-agitation of the milk. There's oxidation that can happen because of exposure to light or certain feed additives. So certainly the feed could be an issue. The flavor itself ranges from sort of like it could be like a burnt plasticky flavor to a metallic flavor. One of the things that we use to train folks on how to detect this is sort of stale crackers, because that's actually an oxidized flavor. So there's a range of potential attributes that can cause that, and definitely exposure to different fatty acids in the feed can cause that.
Okay, let's see. How do we quantify bacteria, CFU per milliliter, in bulk milk? Do we need to culture them in different media or a single medium? Does that work for almost all of these bugs?
Yeah, that's a great question too. So the standard method for doing this is using a media called standard methods agar, as much as that's a kind of a silly way of thinking about it. But anyway, it's essentially just a basic media that's used, and whatever grows on that media at 32 degrees Celsius is what we call the total bacteria count. That's the traditional media way of doing it. I would say, again, that's not really what's used most of the time. Most of the time, we're using this flow cytometry method again, where we're looking at all the individual cells. So there are pros and cons to every method. This flow cytometry method that I'm talking about, where we can line up the bacterial cells through this really, really thin capillary tube and then count them with a laser. It's great because it's really fast. In the traditional method, it takes days to get a result. So when we get a flow cytometry result, then we can get that in a few minutes. It's super, super fast. However, it tends to overcount compared to the standard method, which is the traditional media method, where we put the milk onto an agar plate and we incubate it for 2 days at 32 degrees C, because sometimes bacteria are in clumps and you won't be able to differentiate those clumps on an agar plate. Similarly with like a direct microscopic count, same thing with the somatic cells. We can look through a microscope at a certain volume of milk and then make a determination about the amount of bacteria there, but there are differences. And so there are adjustment factors that have to be used with those different methods in order to accommodate for the change in the type of method, in order to sort of make it in line with what you would find on the traditional media method, which is still sort of the gold standard for that test.
And I was just wondering if you could just tell us a little bit about what's going to happen in that class. You said that for the first 50 people or so that sign up, you're going to send them something. What is that? Maybe
Right.
tell us a little bit about that, because I think that might tempt a few people to
I would
come.
be Yes, I'd be happy to describe that a little bit more. So in our Raw Milk Quality 101, the 2-hour pre-recorded self-paced part of the workshop, we'll be sending out 2 different sort of interactive kits. The first one is going to be milk components. So we allow our participants to actually taste lactose, taste casein, look and smell the different components of milk, as we talk about the importance of each of those components and how they can vary in different products. And then the second one is really introducing the participants to these sensory defects that we can find in raw milk. So we'll send you some stale crackers, we'll send you some cowy and barney odors, and we'll send you some different things to smell so that you can get a sense for what are those attributes and how can they come through in the product. We'll send you some malty product so you can smell that and be able to experience that. But overall, what we're going to go over is the basics, essentially. What types of bacteria are found in raw milk? What's normal? What's not normal? How do they behave differently? And what should you know about them? What are somatic cells? What's important to know about somatic cells and how they are impacted by mastitis. And then talking a little bit more about those quality parameters that we talked about today, digging into those a little bit deeper, so that when we move on to the live virtual workshop on December 16th, you have a really good baseline for accommodating all of the information that our speakers will present on teat end health and how to really optimize teat end health, how to improve somatic cell counts in individual cows, how to make sure that your equipment is operating correctly, and what are the things that you should be checking and looking into if you want to improve your equipment performance. Again, looking at mastitis and all of these things that are influencing these 4 core parameters that we know impact finished product quality.
Okay, great. Thank you. It sounds like fun. So, yeah,
I'm looking forward to it.
Yeah, we have somebody who's looking forward to it. So, yeah,
Great.
I'm really
Hope
glad
to
you're
see you
going
there,
to
Stacy.
come. Thank you so much, Nicole, for this
Thank
really
you.
informative presentation. And we look forward to more webinars from this project over the next couple of years.
Yes, looking forward to it as well. Thanks everybody for stopping in, and I hope to see many of you at the workshop in December.